We've prevented skipping.
The sarcomere shortens from both sides when actin filaments slide along the myosin filaments.
Remember that myosin can also be referred to as thick filaments and actin can be referred to as thin filaments.
The action between the filaments is described as sliding. The myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. Model. The sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten.
We can see the grade and export the grades.